We should go ahead and get started. Okay, so first off, if you're stressed out, completely understandable. If you had the entire term to learn this stuff, it would be one thing, but the fact that you have to condense all this learning down into 4 weeks is a lot. So if you're stressed, just understand you're not alone. This was difficult and is difficult for everyone. So I have an analogy for you. Your brain is like an iceberg, right? And the facts are like penguins on the iceberg, and you could only fit so many penguins on the iceberg, right? So many facts in your brain. So if you try to put too many penguins on the iceberg, other penguins will fall off, right? And they'll fall out of your ear. So the point is, focus on the high yield penguins, all right? Because that's what's going to be tested, and you cannot learn all of this. You're not supposed to learn all of this. You need to focus on the clinical aspects, the rate limiting enzymes. If you want to whiteboard it once just to get the feel for it, that's fine. But we're going to try to point out the high yield stuff that is going to be tested. Okay. Now, that being said, let me share my screen. All right. Now, so here's the plan. We were going to do both weeks, last week and this week, but it's so much. This is going to be an extended, we didn't want to keep y'all too long. Nobody wants to be here for 4 hours. So what we're going to do, first off, I'm on Scrubs, Lindsey's dog mom. Go look at our drives. We put extra questions there too. So you can go look at those. The Mac Daddy question, there's a lot of questions. At some point, you got to just stop looking at the lectures because it's just in one ear and out the other, and just start doing questions. Just like, even if you don't feel like you know it all, just start doing questions. You'll feel better. At least you're doing something different. So this is the plan. Yeah, go ahead.
Just to let y'all know, I did much better on this exam than I thought I was going to. Like, I was having mini panic attacks before the exam because I wasn't remembering everything. Just trust yourself. You do know this information, even if you don't think you do. And it'll start coming back on the exam. So even if you're freaking out the day before the test because you're looking at histo and you don't remember learning it, you'll do fine. You'll be fine. Just trust yourself.
So the plan is today we'll do week 5 stuff and then up through lecture 27. Okay, and it's going to be a lot anyway. And then we'll plan on tomorrow, maybe on Saturday instead of Sunday, since your exam is Monday. We'll cover the rest of week 6 and week 7. I saw week 7 does have, it's a little bit lighter. Maybe there's some nutrition or something. So that'll be easier. So we'll do the 1-carbon metabolism and the heme stuff next week. Just because we don't want to keep you guys too long. All right, so let's look at fructose and galactose. Fructose is going to be basically a disaccharide. Sucrose is a disaccharide of fructose and glucose. So we're going to look at some of the pathways here. Remember that GLUT5 is the one that is related to fructose. The rest are related to glucose and insulin for GLUT4, but GLUT5 is specific for GLUT5 is specific for fructose, so keep that in mind. So we're going to look at this pathway. Here are 2 penguins you should know, fructokinase and aldolase B, okay? And these are going to be, the reason you want to know them is because they're correlated with the disease. They're not going to ask you this pathway and ask you this enzyme here because it is not correlated with the disease. Okay, so the ones in green. So if we look at these, we'll start with this one. This one's not so bad. It's a fructokinase deficiency. They call it benign or essential fructosuria. Typically, it's asymptomatic. You may get symptoms in your teens or 20s with it, but basically, it's not really a large problem. But when you look at these fructose and galactose problems, you should look at the symptomology, right? So the ones with galactose, they have cataracts. Fructose never have cataracts. But also, you should look at the age of onset. Because 1, that'll tell you the severity of the situation, and 2, it'll kind of you can use that to guide you as to like what's going on. Like we'll see those part of the fructose problems. We'll get to the next one. It happens right about 6 months, 6 to 8 months, because that's when fruits are starting to be into the diet of the child. So this is think about teens, maybe 20s. It's usually asymptomatic. Not really clinically relevant. Dr. Mcad really likes to talk about these reducing sugars. So they reduce down and they end up in your urine. So a reducing sugar that's not glucose or galactose, what would it be? It'd be fructose, right? You're not breaking it down. Now, this is the one that is that is worse on the fructose scale. So this is an aldolase B deficiency. So you get that fructose 1 phosphate, and you have a problem with aldolase B. Okay, so like I said, the problem happens. So like I said, no cataracts, right? Because it's fructose, not galactose, or not diabetes, which we'll get to in a second. But they also have this hypoglycemia following sucrose or fructose ingestion. So remember, they'll tell you in the stem, it's about 6 months of age, 8 months of age. Fruits are being added to the diet, right? They're weaning off of breast milk. So this is when this starts to happen. So that's a key, right? No cataracts, 6 to 8 months. You're thinking this. All right, this is from First Aid if you want to read about it. Again, if you're looking for high yield penguins, First Aid is the place to go. All right, now, these are important too. So what happens is with diabetes, you have excess levels of glucose in your blood, right? Your sugar should be around 100 or so, maybe a little less. And sometimes their sugars are around 400, right? So the lens actually has aldose reductase in it, but it doesn't have sorbitol dehydrogenase. For typical people that have normal levels of glucose, this isn't a problem. But if you have excess levels of glucose, you can actually have sorbitol deposits or accumulation in the lens. And that's why you actually get cataracts. So when we talk about cataracts, we're either talking about this in diabetes, or we're talking about it in a galactose problem, which we'll get to in a second. So keep in mind, the problem here is a buildup in sorbitol, okay? So cataracts here, diabetes, we're thinking it's a sorbitol buildup. The enzyme of interest is aldose reductase. They could ask you that, what's the enzyme that converts to the product causing the cataracts. Now, the other one, like I said, in galactosemia, galactitol is the the product that's going to build up. Notice it's the same enzyme. You still have aldose reductase, okay? So it's going to either convert glucose or galactose glucose to sucrose, glucose to sorbitol, and galactose to galactitol, but it's still aldose reductase. So if they talk about a child with a galactose problem or galactosemia, the product that's going to end up in the lens that causes the buildup and the cataract is going to be galactitol. Okay, again, these two enzymes are going to be the ones that cause problems. You can see here. So those are the ones you're going to want to remember. Now, you can see here, so got the cataract, so we're thinking it's either diabetes. For right now, it's either diabetes or it's going to be a galactose problem. So sometimes they call this non-classical, they call it galactokinase deficiency. I see that more often than non-classical. So it's the reducing sugar you're fine. It's not fructose, right? It's galactose. That's what's going to be in the urine because you can't break it down, right? It builds up. Send your questions to Lindsey right now. So and I'll get to or we'll get to him later if we can't right now. Okay, so this one's not as bad as the next one. So galactokinase is sort of like the fructokinase version of the galactose problem. So this one's going to be not as bad, but you still get cataract formation, infantile cataracts. Okay, that's what you're going to think about. Now, this is the one that's really bad, right? The longer the enzyme name, the worse it is. That's not standard, but like multiple choice, you have a long one. But a long enzyme, it's typically the bad one, right? So it's a GALT deficiency, it's autosomal recessive. So this one's really bad. So you start to see symptoms in 2nd to 3rd week of life. And you get this buildup of galactose 1 phosphate, and you get this severe hypoglycemia. Now, what I want you to remember from this, so again, you're going to get this galactose as a reducing sugar, and you get these buildups in the brain as well. So with classic galactosemia, this is the triad you're looking for: liver damage, cataracts, developmental delay. It's so bad that these symptoms start to develop already at the 2nd or 3rd week of life. Okay, so this triad is going to be for classic galactosemia. And you can see this here. This is from First Aid, if you want to go ahead and read through that. Again, this is kind of just a summary when we talk about these reducing sugars or the sugars that show up in the urine. You can see the cataract formation with galactosemia. There's also the fructose problems as well. So that's just a summary slide. Just for completeness sake, we could talk about lactose briefly. Typically, it's a problem with the lactase enzyme in the brush border when you absorb it. So they talk about, and you need to know these genes too, the LCT gene, which is the non-persistent type. Non-persistent means it happens in youth. It doesn't stay through adulthood. And then you also have the MC6. That's more of a persistent problem. I do remember them asking these genes, so make sure you know those. And then if you break it down, so we have the non-persistent one. This is in adults, like I said, and then you can have the disaccharidase, which is kind of a subset of it, right? So it's a secondary problem. So something like celiac disease or some sort of infection like that. It's a secondary infection that causes it. And then the really bad one that you see is congenital lactase deficiency, right? So any sort of milk, lactate, you have to give the child lactose-free milk. So this is very severe. They end up vomiting a lot and getting really sick. Okay, so yeah, again, it's a lactase deficiency. They get a lot of osmotic diarrhea. Osmotic meaning watery, right? Okay, and again, it's that brush border enzyme. Secondary, like I said, it's a secondary problem. You can get it, sometimes it's rotavirus, some sort of intestinal injury, some sort of problem with that brush border, right, when you absorb it. And when that happens, you get this secondary lactose intolerance. You can't absorb it. Again, from first aid, if you want to go ahead and read through that. All right, let's move on to fatty acids. These aren't as bad as they need to be. Okay, so remember, tags are going to be the storage forms of fat. You can see here, just to point out, so this is saturated, right? It has no double bonds. This is unsaturated. The reason monounsaturated fats are better for you, or even the cis bond here, is because it puts kinks here. And so it's more fluid. When you have saturated fats or you have trans fats, which the trans would bend the double bond the other way, these strands end up being tighter. So they end up being solid in solution. So they tend to form plaques more. So that's why saturated fats are bad, and specifically trans unsaturated, because they have more of a solid form. But these cis forms and this monounsaturated, that helps to keep fluid, so they don't tend to make plaques, they don't develop, or they don't recruit cholesterol as well. So that's just a little tidbit. Right, so when we talk about this, remember we did this last time, the on is sort of the well-fed state or the insulin-based state. After you eat, you want to store what you can store, right? So you're going to store fat. So this is when we're going to go through this storage form of fatty acids. You definitely, NADPH here is one of the the reacting equivalents that you need for this process. Remember, NADPH is only used in a few places. This is one of them, along with, I believe it's cholesterol biosynthesis as well. So you can get that through these pathways, the pentose phosphate pathway, the malic enzyme pathway. So basically what we're going to do here when we form these fatty acids, these tags, right? So you start out with this acetyl-CoA. And first of all, let's just point this out. Acetyl-CoA can't get into the mitochondria, so you have to convert it to citrate. Now, once you have a lot of citrate, you can use citrate lyase to convert acetyl-CoA from acetyl-CoA and oxalic acid. Yeah, right. So you can convert it back. Use this citrate in cytosol to form acetyl-CoA. Okay, right. So it's just to convert citrate back to convert acetyl-CoA to form it. Yeah, it's used citrate to form
acetyl-CoA.
Thank you. Yes. Okay, so it's going to convert. It's just confusing because I was thinking in the mitochondrial membrane. Anyway, citrate lyase is going to convert citrate to acetyl-CoA. Thank you. And okay, so we look at this process. So the enzyme of interest is acetyl-CoA carboxylase, or ACC. This is going to be your rate-limiting enzyme. This is going to be an enzyme you want to remember. So acetyl-CoA is a 2-carbon molecule. You're going to use CoA carboxylase to add a carbon to it. then you're going to get malonyl-CoA. So that's that rate limiting step. Then from there, all you're going to do is add a bunch of acetyl-CoAs to it, right? Carbon change, you're just going to stack them up on it. And that's the process of making it. So again, you're going to have acetyl-CoA, that rate limiting enzyme. Acetyl-CoA carboxylase is going to make malonyl-CoA. That's a 3 carbon. And then you're just going to add a bunch of acetyl-CoAs to it, right? Just add them on till you get to palmitate, which is 16 carbons. And then you're going to start going into the triglyceride synthesis, right? You're going to put that backbone on it. So definitely remember acetyl-CoA carboxylase and malonyl-CoA, right? That is the end product or after that rate limiting enzyme step. Okay, definitely need to know that.
Is this
okay.
in the cytosol?
So it's actually in the mitochondria.
Oh, mitochondria. Okay, thank you.
Yeah, right. So you convert. Why does this looks like a mitochondria? No, that can't be a mitochondria.
It's all in the cytosol except the first reaction. So you have to take citrate out from the mitochondria because acetyl-CoA can't pass the mitochondria. And then once you get citrate, you're going to cleave it to acetyl-CoA on oxaloacetate and then do fatty acid synthesis.
Okay, great. Thank you very much. So yeah, so the citrate lyase, once it gets back into the cytosol, we'll convert it back to acetyl-CoA. Thank you very much. All right. So like I said, acetyl-CoA carboxylase is going to be that rate limiting enzyme for this process. It starts off as proteimmers and they come together in the high concentrations of citrate, and it turns into this polymer formation. So for that rate limiting enzyme, you're going to take a 2 carbon acetyl-CoA and make it a 3 carbon. Okay. So fatty acid synthase is going to basically add those acetyl-CoAs to it, right? So you're going to keep adding them on until you get to that 16 carbon chain. And so that's when you want to remember too. And then you get the palmitate, and that's kind of where it maxes out. And then you go to the triglyceride synthesis. And I did kind of mention, right? So you could see this is a monounsaturated fatty acid. So if you put this in the membrane, it'll make the membrane more fluid, right? So when it's floating through your bloodstream, it tends to be fluid. Whereas this, or a trans fat, which would bend it back to make it more of a straighter formation, it'd be more likely to be solid. So that's why they tend to develop solid plaques in your arteries and stuff. Okay, so remember the trick. These are essential amino acids, right? So remember the trick. If you go backwards, 1, 2, 3, 4, 5, 6, and find the N. The N is omega-6. 1, 2, 3, omega-3. So find the N. That's how I remember which one is which. All right, again, we want to.
Blew my mind.
Yeah, you never heard that one?
No, dude. I had the longest trying to figure out because like I just had to hard memorize it, but you just okay. Thanks.
Yo, whatever works, man. All right. Okay. So what happens here when we talk about feedback inhibition? Now the next one is more important than this, but what happens here is after you make this 16 carbon palmitate and you get to this process, this fatty acid CoA. If you don't have the products to start making triglycerides, this is going to build up. So it's going to feed back and tell palmitate to stop forming. So we're going to just kind of stop forming because we don't want to build this up if we can't fully make triglycerides. Okay, so keep that in mind. This long chain fatty acid acetyl-CoA, it will help to slow it down. And then fatty acyl-CoA, yeah, right. So that's going to be what causes the feedback. Now I wanted to point this out. Okay, it's coming up. So beta oxidation is going to be basically breaking down these fatty acids, right? So let's see. Yeah, so here, this is the enzyme, this is the rate limiting enzyme, fatty acyl-CoA synthetase, right? So it's going to break down till you get these fatty acyl-CoAs. So this is going to be this activation step. And you could see that here, again, rate limiting enzyme, and you're going to use this carnitine shuttle to get it across to the mitochondrial matrix, okay? So then once you get these acetyl-CoAs, you're going to finish undergoing beta oxidation. So just think of it as like when you're building these tags, you're just kind of just putting on these 2 carbonyl-CoAs as you go along, right? When you're taking them off, you're basically taking them off as acetyl-CoAs, like 2 carbon chunks as you go. Right. So, and this kind of illustrates it here. You kind of keep going through this step and you're just knocking off 2 two carbons or acetyl-CoAs as you go. And that's the process. This is what I wanted to point out, which is very important. If you have a buildup of malonyl-CoA, right, after this ACC enzyme step, excuse me, if you have a buildup of that, that is going to tell the fatty acids to not break down, right? Because this is kind of one of the end products or close to the end product of beta oxidation. So if you already have a lot of malonyl-CoA around in the cell, you're gonna stop breaking stuff down, right? So definitely remember this, that malonyl-CoA is gonna inhibit the process of beta oxidation. Definitely keep that in mind. Okay, and it may be worth just running through this, just kind of working out how many ATPs, but you could see, look how much ATP you get from breaking down, fatty acids, right? It's a ton of energy. So it's important to kind of know where these steps break down into. Okay, now let's get into the diseases, the most important stuff. So the Mcad is going to be the most specific one. That's going to be the 10 to 6 carbon, but there are problems with 16 carbon as well, which we'll talk about. So Mcad deficiency, the thing you're definitely going to want to remember with this category, specifically MCADs, is that you're going to get hypoglycemia and hypoketosis. Besides the systemic deficiencies that we're going to talk about in a second, I'm pretty sure this is the only situation that you're going to get both hypoglycemia and hypoketosis. Don't quote me on that, but I'm pretty sure, like on the test, whenever I saw this, was like, it's got to be Mcad deficiency. So we have a problem breaking down these 6 to 10 carbon chains. And you can see that here, hypoglycemia, hypoketonemia, right, or hypoketosis, right? And definitely remember this, they'll put that on the test. Patient comes in with dicarboxylic acids in the urine. And so you're gonna know that's Mcad. So you're basically, when you look at the pathway, like you either go down beta oxidation, you do this omega oxidation. And if you can't do beta oxidation, you have to go to omega, right? So that's why you get these dicarboxylic acids, it's inevitable. All right, now these systemic carnitine deficiencies are here. So these will show up with hypoglycemia and hypoketosis. Basically, this falls in the same category as the Mcad deficiency. Now, you also have ones that are specific to the muscle as well, this muscle carnitine deficiency. Now, CPT1 falls in this category, and I have a summary slide in a second. So this is going to be a systemic form. CPT2 is the myopathic or the muscle form. And you could see this here. So any of these that fall into this systemic fatty acid oxidation disorders, MCAD, CPT1, or carnitine deficiency, you're going to see this hypoglycemia, hypoketonemia, right? Hypoglycemia, hypoketonemia. The myopathic ones, you'll probably see an increase CKMM, right? Because it has to do with the muscle specifically. So keep these together. It would be difficult if they put multiple of these in as answer choices, right? Because they kind of all sound the same. But if you see dicarboxylic acid, hypoketonemia, hypoglycemia, definitely think Mcad deficiency. Okay, Jamaican vomiting sickness, they like this down on the island. Well, they like talking about this down on the island, but basically it causes Mcad deficiency, this enzyme that works there. So it'd show up with similar symptoms. Okay, just to point out that you can end up with odd chains, this propionyl-CoA. Remember, anything that's a carboxylase uses biotin as a cofactor. And this is one of the 2 places where you're going to use B12, this methylmalonyl-CoA mutase, which we'll talk about the deficiency there, which isn't very common, but we'll talk about it when we get to Dr. Sobering stuff. So this is just, I don't know if I'd really remember that. I may just remember the enzymes, right? Just what the cofactors are for this odd chain fatty acid breakdown. Now, hormone-sensitive lipase is important. Remember, lipase, you're breaking down, it's an enzyme to break down lipids, right? Lipolysis. And so that's what you're going to think about. So at what, when do we want to do this, right? You're going to be low in insulin. You're going to want to mobilize glucose. You're going to want to increase your blood glucose. So this is the point where you're going to go use hormone-sensitive lipase. So this hormone is on glucagon's team, right? It's going to want to increase your breaking stuff. So keep that in mind when you go forward. If you want to see how it works, the synthesis and the beta oxidation, I like how First Aid kind of puts it here. you can see the different important steps, and they have some of the easy ways to remember stuff. All right, let's talk about ketones, right? So Zellweger syndrome is one you need to remember. When you hear a Zellweger, you need to think about peroxisome. It's a peroxisomal problem. So you don't have this proper peroxisome biogenesis. So what happens is you can't break down these long chain fatty acids. And so that's what they'll probably say. They'll probably say they have Zellweger syndrome, or they can't break down very long chain fatty acids. And they'll ask you, where's the problem? And it's in peroxisomes, right? They get a lot of neurological manifestations, and it's also in the liver as well. So liver and brain. A subset of that, this is not as highly tested, or I don't believe it's as common, but it's Refsum disease. So this is specific for alpha oxidation. It does take place in the peroxisomes, but it's more of a specific problem, this alpha oxidation problem. So phthalic acid is something you want to keep out of your diet. Okay, so let's talk about ketones. So remember, we'll break down beta oxidation. Remember, we're going to be cutting off the triglycerides, the tags, into different acetyl-CoAs, right? So ideally, we go to the Krebs cycle. But if for some reason, and I'm going to point this out again, what this branch right here is going to be determined by NAD or NADH. If we have too much NADH, we're going to shunt towards the ketone body synthesis. So that's an important precursor, because if you don't have, you need NAD to go to the Krebs cycle so you can push through to make NADH. So when we talk about this ketogenesis, the NADH is going to be that primary mechanism to tell you to go make ketones. Right, so ketones are going to be in that fasting state. It's not an ideal situation, but if you're fasting and your brain needs energy, you got to make ketones. It's just is what it is. So 3-hydroxybutyrate is going to be the one you are most interested in. That's the one that's going to spike the most. Acetoacetate's formed, and then acetone can be formed from that. So here's a penguin for you. This is going to, now remember, this is for synthesis. Don't get them confused with the breakdown. So when we start making ketones, mitochondrial HMG-CoA synthase, right? You're synthesizing these ketones. So acetoacetate forms here. It could break down to acetone or beta-hydroxybutyrate. So definitely remember this one. Now, this is a perfect example. I probably wouldn't draw this whole thing out. I would just say this one's really important. Okay, you got to pick your battles here. Okay, now ketones, again, not an ideal situation. The brain has to use it at times, but ideally you want glucose. But again, we're going to look at that NAD. My dog's snoring. She's bored. Sorry. Don't blame me. So we're going to look at the NADH and the levels of that going forward, because that's going to kind of point out what we do. So now let's talk about when we utilize these ketones, when we break these down. Again, look what we're forming again. We're forming acetyl-CoA. This is going to be like that it's a 2-carbon. It's a very simple structure, right? So when we want to break down these carbon chains, that's how we do it. So when we break down ketones, succinyl-CoA, acetoacetate-CoA transferase, also known as thiophorase, is going to be the key enzyme in breaking it down. Again, probably wouldn't want write this whole thing out, but if you want to, go for it. Now, again, this would make sense, right? Why is it only in the peripheral tissue? Because you're mobilizing these ketones. This is the last ditch effort to use any energy you have, right? So the liver is going to send it out to peripheral tissues, and that's the place where you're going to break down ketones. All right, let's see. And you can see that here. Look, so we start with fatty acids, right? We're going to start breaking them down. We're in a state of starvation or fasting, right? We're breaking these down. So we're going to start making acetyl-CoA. So we're going to cut them off, right? And then we're going to get to a point where there's too much NADH, okay? So you're going to go one way and you're going to break down the NADH to NAD over here. But this increased NADH is going to shift us to this pathway to making ketone bodies. If the opposite were true and you had NAD, then that's fine. You can go through the Krebs cycle. You can, go through aerobic glycolysis. But the problem here is this is a very important molecule, right? So we need to go toward the ketone body. It kind of just signals to the body that you're in more of a starving formation. Now, this is interesting because what you see here is after the first 24 hours, you've basically used all of your glucose stores, right? So your glycogen's wiped out in the first 24 hours. So then you need to start making new glucose. So you go through gluconeogenesis. And this process of being able to make new glucose will keep your glucose steady for 40 days or so, which is crazy when you think about it, right? Because you're out of stores in the first 24 hours, but you can actually make it and keep it steady, but you're still at a low level. You're at like 40 or 50, your glucose levels in your blood. So you're not feeling too well, but you could see as time goes on, you're going to be making a lot of ketone bodies. And remember, again, that beta hydroxybutyrate is going to be that important one that we talk about. So now this is super important. You could star this slide because this is something really important clinically. So DKA, diabetic ketoacidosis, we're thinking about type 1 diabetics. So let's look at the situation here. Kussmaul breathing, those are deep labored breaths, right? Fruity breath should be a red flag immediately. That's acetone coming off the breath. That's why they get that fruity breath. But their plasma glucose is 400. I think Dr. Mpaji put it, she had an elegant way of putting it. It was like this, like starvation in a place of excess, or in a situation of excess. You have tons of glucose. Why are you in a starvation situation? And that's simply because they don't have any insulin, right? So even though your plasma glucose is this high, you don't have insulin, so your body's still starving. You can't get that glucose from the blood into the tissues, right? So you can contrast this with type 2 diabetics. If they were in a fasting situation and they go through a similar, it's different. It's a different ketone process or ketogenesis process. But if you were to look at these separately and say they're making ketones, what's the difference? How do you know if they're type 1 or type 2? You check the plasma glucose. So in type 1, remember, they're not making insulin at all. Okay, so that's why the glucose is so high, but their body's starving even though they have excess glucose. Whereas a type 2 diabetic, it's probably some sort of insulin resistance that's forming, so their glucose will be low in this situation of ketones. But by all means, if you see DKA for the test, you're thinking type 1 diabetics for sure. They get this anion gap, metabolic acidosis, so the ketones don't count towards the anions, right? So it forms this gap. And this is interesting too, right? So if you have all this glucose, you wouldn't expect hormone sensitive lipase to be breaking stuff down, right? To be making ketones. But the problem again is there's no insulin. So the body kind of still feels like you're starving, even though you have all this glucose. And the problem is you don't have any insulin. Okay, cool. So I think that's all of that. Yeah, but make sure you know these symptoms. That'll help. Right, again, I just wanted to point out this hormone sensitive lipase. It's on glucagon's team. It's going to mobilize or break down lipids, right? So you'll see that here. This diabetes, you'll get ketonemia, ketones in the urine, stuff like that. Hagma is what they call this, high anion gap, metabolic acidosis. All right, that wasn't so bad. Do you want to go ahead, Lindsey, and then we'll take a break?
Yeah, sure. We could do the histo and then we'll go, and then we can do the glands if y'all want to do that in a bit.
Are you want to? We didn't take too long with that. We could just do them now and then take a break, and we'll be about at the hour mark.
Okay.
All right.
Okay, let me get this. Okay.
So Lindsey will go ahead and do the histology with you guys, and then we'll take a break, and then we'll go into this last week's material. They're asking about your slides, Lindsey. We'll compile them compile them after and repost it.
Okay, which one? This is one. Okay. So. Okay. So the DM histo, again, I put what you should really focus on, like the major penguins that should be on your iceberg hanging out. So, okay. The tongue, this wasn't a huge thing, but you do need to know the difference between all of the different taste buds and where they are. So you need to be able to look at this and know which ones you are looking at. So of course, the filiform, they're a little more, I think they look like spiky. Fungiform, the papillae, and then the circumvallate papilla. And make sure you know the location of all of these. Some of them are dispersed. So the filiform are going to be distributed over the entire surface. Fungiform are scattered among the filiform. Folate are on the lateral edges, and then circumvallate are on that that border. They like to focus on that, but this wasn't a huge thing. Don't stress about this, but do definitely know how to distinguish them if you're looking at one of these pictures. Now for the rest of this tract, make sure you know the big differences between each part of the digestive system, because that's what's going to get you focused on each section. So I put this on here. So if you see something like this, so you have, this is your lumen right here. You have your epithelium, lamina propria, muscularis, and you have your glands down here. Make sure you just know what this looks like. So if you see it, you can know, yes, this is my esophagus. This is the esophagus. And of course, if there is a junction, they love to focus on the junctions, because what can happen at the junctions?
Metaplasia.
Yeah, metaplasia. And so anytime that can be pointed out, they will probably put it on there, because especially at the gastroesophageal junction, when you have prolonged acid reflux, that's when you can get Barrett's esophagus, and that's when you can get cancer. So just please recognize that junction. Yeah, metaplasia is reversible. So this is just talking about Barrett's esophagus. Again, if they do this, they will talk about it with respect to metaplasia. Okay, stomach, you do need to know the different regions and the glands in each of the regions. That's really important. So the the cardiac region has cardiac glands, fundic region, the gastric glands, the pyloric regions, the pyloric glands. That is really high yield. Make sure you understand where each of those are, because they can ask questions about different parts. And then this isn't part of histology, but do make sure you know the function of each of these. So the cardiac region is where you is where the food bolus first enters, and it's kind of like the reservoir. And then as it moves through the stomach, it has different functions. So make sure you do understand the 3 parts of the stomach and then the glands in each. So cardiac region, you need to understand this is short pits, short glands. So you'll see the pits are right here, the glands are right here, so they're pretty short. Whereas the pyloric region are going to be long pits and short glands, so it extends much deeper. Fundic region. This is super high yield because of the cells in this region. So please make sure you do know the cells. I have them in the subsequent slides. So you have in the fundic glands, yes, you need to know these. So hydrochloric acid is by the parietal cells. Pepsin is the chief cells. You have the mucus. And then you have intrinsic factor. You need to know all of these. You need to know how to identify the cells. And you need to know, so identify the cells. And then when you identify the cells, you need to know what molecule what enzyme is there, and you need to know what it does, because we had a couple questions where it pointed to one of the cells, and it was like a third order, fourth order question where you had to know the functions of everything in this. So big thing here, intrinsic factor, this you always associate it with B12. And remember, B12 absorption is in the distal ileum. This number 4 right here is very high yield, and they can come up with multiple questions in this information. So I put all of these side by side here. So to look at these, hold on, I'm going to move this out of the way. Okay. So mucus cells, mucus neck cells. So you have the surface and the mucus cells. So these, of course, this just is the thick viscous mucus that lines the gastric pits. Parietal cells. Remember, this is HCl and intrinsic factor. Remember, super, super, super important. So the parietal cells, they're big and pink. They kind of look like a sunny side egg to me, but they're really eosinophilic with respect to everything around them. And they're pretty large too, with a big nucleus. So make sure you do know how to distinguish these. And then the chief cells, these are a little harder to pick out because they're a little more basophilic, but they're not so basophilic that they jump out at the slide to you. But they're fewer in number, they're towards the end, whereas the parietal cells, you see them kind of up in the neck. Oh, hold on. Wonder if I can. Sorry, I was trying to find the pointer. Oh, here we go. So the parietal cells are more in the neck, whereas you see the chief cells down towards the bottom. So make sure that you can distinguish between these two. Okay, that's weird. I don't like it. Huh. Again, another junction, gastroduodenal junction. Pyloric sphincter. I vaguely remember this picture being on our exam. But this is, so you would need to know this and what the junction is. So you'd need to know that this is the stomach, this is the pyloric sphincter, this is duodenum, and this is the gastroduodenal junction without it being labeled and with just a clinical picture. Peptic ulcers, I put this in here for completion's sake. A lot of peptic ulcers they're going to talk about, and Brady talked about this last time, the ruptured peptic ulcers, any way you can see that presentation. So if you see a ruptured peptic ulcer, you can see air on an x-ray. You can get content spilling into the posterior of the stomach. So this is more for completion's sake. Small intestine. Now we're getting into if you learn this well, it's really easy to distinguish because each of these parts, duodenum, jejunum, ileum, and then we get into the large intestine, each of these has a very distinguishing factor. And if you know the distinguishing mark, you can pick it out really easily. But if you don't know the distinguishing mark, you're going to sit there and you're going to bang your head against the wall. And this keeps coming back. This is the thing that kept coming back on every single exam for the cumulative stuff. So small intestine, first, please know the difference between the plicae and the villi and the microvilli. This was something that they hit on. So this whole structure right here, PC, this is the plicae. And then you can see the individual villi in the plicae. And then the microvilli is, of course, going to be on the border.
Wait,
so
can you go back real quick?
Yeah.
So the plicae is this whole invagination, right?
Mm-hmm.
And the villi, those are like the finger-like looking.
Yes.
And where's the microvilli you said?
Microvilli you can't appreciate on here because it's too zoomed out. Microvilli are going to run along the villi. So each one of these specializations is going to increase the surface area. So you'll notice that the villi are invaginations of this epithelium, these individual villi, this plicae. Okay, sorry. Starting over. Plicae are the invaginations of the submucosa. Villi are of the lamina propria, and then the microvilli are on the very surface.
Oh, I see. Thanks.
Yeah, that's the brush border, and we'll talk about that in the next slide. So this on the upper left, the plicae, I'm pretty sure this was on our exam, so make sure you understand where this is. So it says most numerous in distal duodenum and jejunum. I don't think that was a big thing on the exam, other than just knowing what these were, but make sure you do understand this. And then, this keeps getting in the way. The villi, central lacteals, that was a thing. I remember that being a thing. And then the villi, this is the brush border that we've been talking about with the enzymes. So if you have a deficiency, an enzyme deficiency, they talk about it with being in the brush border, because as the food bolus moves through the intestines, the microvilli with the enzyme, that's how it's going to react. But if you lose the brush border, you're not going to have those enzymes there. And so that's why one of the reasons, not the only reason, one of the reasons why you can have these enzyme deficiencies or intolerances. Intestinal glands, this is very important. So this is the crypt of Lieberkühn. Okay, now we get into the cells. These are super high yield. So the enterocytes. So you need to know if a cell has a specialization. So in the enterocytes, they have the microvilli, the tight junctions for absorption, and then the lateral plication. So you need to know that the lateral plication increases the lateral surface area. So the specifics of all of these, you do need to know those. And the goblet cells, know what they look like. And they are the I can't talk this morning, mucinogen granules at the apical cytoplasm. So know what they look like. Panna cells, these were the most high yield. The biggest thing is the alpha defensins. That's the biggest thing. Know what they look like. Know the alpha defensins. This kind of got cut off at the bottom, but it's the lysozyme, which is an antibacterial enzyme. And then enteroendocrine cells, these are at the base. The big thing is that the most active regulators of the physiology. So CCK, secretin, GIP, motilin. Duodenum, Brenner's glands. Every time you see duodenum, you need to think to yourself, Brenner's glands. Those need to be heavily associated in your mind. Jejunum, we're talking about the plicae. So, okay, earlier I said these weren't specific to an area. They are. I apologize. Jejunum, always think about these plicae. So,
can you go back one slide real quick?
Yes.
So look, this Brunner's gland came up, I think, on the exam and on the cumulative. Like I thought that this was the large intestine. I got it wrong.
Yeah.
I thought those were like goblet cells in the large intestine. So make sure you can differentiate these Brunner's glands. It's very specific. So that was definitely on our exam.
Yes, 100%. Jejunum, you have the plicae. Ileum, you have these Peyer's patches. So make sure that they can give you the longitudinal section or the transverse section, and you can look and see that these are Peyer's patches. And again, absorption of B12, this is very high yield. You do need to know that, but Peyer's patches, you always want to think ileum. Celiac disease, I think they asked us one question on either celiac or Crohn's disease. Look over this. The big thing here with the celiac is the blunted villi. That's the big thing here. And you can see it on here. You don't see that feathery appearance. You more see the blunted appearance. So that's what that's talking about. Large intestine. So the tinea coli and the Hostra coli, these are the two big things that you are going to be able to identify the large intestine versus the small intestine. So that tinea coli are these longitudinal bands throughout the large intestine, whereas the Hostra, those, if you, I don't know if you've gone through this in the physiology yet, the Hostra are what kind of move along. And if you look at an x-ray of it, you look at barium x-ray of it, it's almost like, I guess you could call it beads on the string, like really big beads on the string that move along. But the tinea coli runs longitudinal, and the Hostra are the, they're very visible, the pouches, I guess you could call them. And you'll get into that a lot with physiology. But also the thing, you can see this tinea coli, these longitudinal sections, you can, you can see the longitudinal band on that transverse cut. But those are very characteristic of the large intestine. And then this is what Brady was talking about. These, because I did the exact same thing Brady did on our cumulative stuff with the Brenner's glands in the small intestine, because I thought it was a large intestine because I was thinking about this right here. So make sure that you can put those two side by side and you know what the difference is between them. So a lot of this was more first and second order in identification. Like they'll give you the patient presentation, but it's going to be more off of the identification of the actual slide that they attach to the question. So please make sure you can put them all side by side and you don't forget them. These are the easiest to test cumulatively. Muscularis externa. This is, so I think we had this upper right picture on our exam, but that is, it's just, if you see this picture, you just know what it is. Appendix, this was a big thing. The lymphatic nodules is a really big thing in the appendix. So to be able to identify this from every other part. Anal canal, this you do need to know the different sections of this. Remember the remember below the pectinate line, above the pectinate line, when you get to the anal canal, there are different sections. I didn't hit this too hard. They more hit it hard in small group and in lecture, but do make sure that you can look at this and you know the different zones. I think we maybe had one question on this, but make sure you do understand this. I put this on here more for completion's sake. The congenital megacolon Hirschsprungs, we talked about this last week. When they put this on your exam, they're probably going to put an X-ray on your exam or a CT. And if you see this picture on the right, this cartoon graphic, you'll see this extremely enlarged sigmoid colon right here. And so you're going to see this on an x-ray and it's going to look like it fills up essentially the whole abdomen. So that's what megacolon is going to be. Again, autonomic ganglia. This is a failure to develop the myenteric plexus. So you don't have those peristaltic movements, so it's going to build up in that sigmoid colon and that descending colon, and then that's why you have the severe dilation. Okay, salivary glands. Brady, I don't know if you got my message. When I tried to like transfer this over, some of my gland slides got really messed up. So we can take a break here. I'm going to like rearrange those so they're good.
That's fine. Let's take a 5-minute break and we will we'll come back after that. Yep. Yeah, for sure. All right, let's talk about cholesterol. It's real fun. All right, so what is cholesterol used for? It is a precursor for vitamin D. It's also a precursor for steroid hormones, which you remember learning that. And it's also used in bile acid synthesis, and the bile acids are going to be super important in transferring or solubilizing the cholesterol. It's also used in the brain. So when we look at cholesterol, here we go. So let me just point this out just for clarification's sake. So pyruvate is going to get shuttled into the mitochondria. Acetyl-CoA isn't able to cross into the cytosol back. So you have to convert it to citrate, then citrate is shuttled across. Then you can convert it to oxaloacetate. But like we were talking about earlier, this is that branch port, and you can start to make oxaloacetate as well to go back into gluconeogenesis. And with the increase in ADH, you can make acetyl-CoA. Now, you could all go to make fatty acids, but you can also end up making cholesterol. So enzyme of interest, this is your penguin here, HMG-CoA reductase. Now, what I did, if you feel like writing, as much fun as it is to say squalene, if you want to write all this out, you can, but I literally just remembered this, okay? HMG-CoA reductase right here. This is what's important. This is the rate limiting enzyme. This is the enzyme where statins work, okay? That's really all you need to know for this. Okay, there is one disease. So if you see here, these bunch of lines right here, this is a multi-step process. So the last step can actually can cause a problem. This Smith-Lemli-Opitz syndrome. So what you see here is this buildup. You don't have this DHCR7 gene. So you have this buildup of the precursor 7-dehydroxycholesterol. And so you end up having decreased cholesterol. Now, classically, you can give cholesterol administration. This will help these kids, but it actually doesn't help with the neural development. So they end up having some neurologic problems. Why they get, I would love to know why they actually get some sort of syndactyly here. That just seems like it's so random for this to happen, but that's something. This wasn't super high yield, but it's just one of those things. It's clinical, so I wanted to discuss it. All right, so when we degrade cholesterol, remember we move it from the liver and we store it in the gallbladder when we convert it to bile acids, right? So this is the one, you do have microorganisms, but primarily we're going to talk about this in regard to the bile acids and bile salts. Okay, so when we talk about bile acids, what are we talking about? So primarily they'll eliminate the excess cholesterol. You use cholesterol to make these bile acids, and you'll be able to solubilizing them as well. So ideally, you would not precipitate them. Obviously, this is a huge problem, though. If you have too much cholesterol in your diet, you tend to make gallstones. Remember, the way to remember that typically it's female, 40, and fertile. I didn't make it up, but those are what goes with gallstones. So that's typically the presentation. Yeah, and so you can digest tags as well. It's an emulsifier. And of course, remember fat-soluble vitamins. So if we want to talk about a clinical aspect of this, when we talk about something like cystic fibrosis, which will come up again, well, we have this blockage, this gallbladders, and we don't get the bile acids in there. Remember, we have a problem with this absorption of fat-soluble vitamins. This is classic for a cystic fibrosis problem. Okay, so the proper term for gallstones is cholelithiasis. Lithiasis, yes. So you look at how many gallstones are formed here, right? So a lot of times by the time it's presented clinically, there's a lot that is formed, or it's got some sort of, it's lodged somewhere in the bile duct, the bile tract. So remember I mentioned before, I think next week y'all will get into the lab values. So remember at the lab values, depending on where the stone is lodged, could kind of tell you, or the lab values can tell you where the stone is lodged. If it's specific to that cystic duct, maybe your GGT and ALP is elevated. If it's up here, maybe your ALT-AST is up. And if it's down here blocking everything, then including your pancreatic duct, you may see that the amylase and lipase is up. So it can kind of tell you how bad it is. Remember, and we'll talk about this in a bit, CCK is going to be what contracts the gallbladder and relaxes that sphincter voti to allow the bile to come out. So a lot of times they'll talk about a patient who eats a fatty meal and they get some sort of cramping in the upper right region. You know, that a lot of times that is a problem because CCK is going to help contract it, but if you can't release the bile, then that's a problem. So you get some pain, some visceral pain. So they also get steatorrhea or like, and this is due to this lipid malabsorption. So remember that the bile acids help to with lipid absorption. So if you don't have proper bile release, you can get lipid malabsorption. So you can get steatorrhea, which is basically fatty stools, which is another thing that happens with cystic fibrosis as well. So they may indicate it as foul smelling, but that's related to the lipid content. Okay, again, you can kind of see where it's lodged here. So basically the problem is going to be, for the most part, related to cholesterol. Either you have decreased bile production or you have increased cholesterol production. Either way, you're going to get these precipitation of cholesterol stones, okay? So that's typically what you're going to look at here in regards to this cholelithiasis. Okay, let's see. And then again, remember, we're in a well-fed state, insulin's on. So insulin's going to favor the formation of storing fatty acids and making cholesterol, right? We're building stuff because we're in the storage form. We're well-fed, our glucose is up, so we want to store cholesterol. Glucagon, again, is going to be the opposite. It's off. We want to mobilize stuff. We want to increase our blood glucose. So we're not going to be worried about necessarily making it like we would be in a well-fed state. Very important, statins. They're a structural analog of HMG-CoA, which should tell you they are competitive, right? They're a structural analog. They bind to a similar site. So it's a reversible competitive inhibitor, just in case they ask that. So it's going to block the formation of, what is it, malvinate? Yeah, or something like that. Malvinate, I think is how you say it. So yeah, so that's the idea of decreasing your cholesterol. That's the first line treatment, the statin treatment. To decrease cholesterol. All right, let's talk about the amino acids and urea cycle. I know this is like a burden to try to go through. It's a little difficult to understand, but I'll try to break it down as well as I can. So nitrogen is going to form ammonia. Ammonia is a problem specifically in the brain. So the question is, how do we get rid of all this ammonia? Well, what we can actually do is form urea. You form urea in the liver, and then it is shuttled to or sent to the kidney, and then you can get rid of the urea. And this process is the way of getting rid of the excess nitrogen that's building up or the excess ammonia. So you can see that if you have a liver problem, you may end up with some sort of urea problem, or you may end up pushing too much urea into circulation. But if you have a kidney problem, or renal failure, you may have increased urea as well because you're not able to eliminate it or get rid of it. So one of the main things you want to remember here is glutamate. Glutamate is going to be what's used to mop up these excess nitrogen or get rid of this ammonia. So it's like a storage form for nitrogen. So you can actually store it in glutamate, and you could actually add another urea or another nitrogen basically to form glutamine. So this is a way of using amino acids and shuttling this to get this toxic urea, or I'm sorry, this toxic ammonia out of circulation, and you can store it here. So cystinuria is basically cystine stones. This is a good clinical application. So there's a problem reabsorbing it back into the urinary system. So basically they end up precipitating. So you get these cystine stones, and oh, okay. And so it's common in children as well. What I wanted to point out is this here. They cause these hexagonal stones. So this is a good buzzword for you to remember. That is indicative of cystine stones or cystinuria. Hartnup disease is another thing you should remember. It has a defect in transporting tryptophan. So basically what they get here is with the tryptophan deficiency, you tend to not make NAD. So this is actually comes out to being a niacin deficiency. So you get pellagra. Pellagra is basically a niacin deficiency. So with Hartnup disease, you should put Hartnup and pellagra and niacin together. Now, what are the signs? They say either 3 or 4 Ds of pellagra: diarrhea, dermatitis, dementia, maybe even death. So this is correlated. So keep Hartnup, tryptophan, pellagra, and niacin. Those are all kind of go together. Okay, so now, in regards to this lecture, what is the overall picture? The overall picture is, how do we get rid of nitrogen? How do we store it? How do we get the ammonia out of the blood because it's toxic? So let's look at this. So what I did here is let's follow the nitrogens around. Okay, so alanine is going to have a nitrogen. So let's say, so if we go this way, remember glutamate is a storage form of nitrogen as well. So if for some reason, alanine is going to store this nitrogen, if we want to get down to pyruvate, maybe break down the alanine and get it into the TCA cycle and get it to pyruvate, we need to transfer this nitrogen. So you use alpha ketoglutarate and you transfer the nitrogen to glutamate, okay? And then you can convert alanine to pyruvate. But again, what you ended up doing with that nitrogen is storing it on glutamate. So ALT, that should be familiar to you, that's in the liver. And transaminases, that's going to use PLP or vitamin B6. So this is important, but just understand that the gist of this whole situation is that we're trying to get rid of the ammonia, so we have to transfer it around until we can get it to urea, till we can get rid of it. Okay, same thing here. Let's follow the nitrogens. Aspartate can hold a nitrogen, asparagine can hold 2. So you can see here, if we want to end up breaking down, so let's say we want to break down asparagine, we want to break down aspartate, we want to feed it in to oxaloacetate for gluconeogenesis, right? Well, that would mean we had to form ammonia, which is toxic. You can even transfer the nitrogen to glutamate, okay? So the idea is you have to shuttle it around for the purpose. If there's a lot of nitrogen or a lot of ammonia, you may end up storing it as glutamate or glutamine for the time being, but eventually you want to be able to get rid of it, okay? So there's a common theme here of being able to shuttle around all these nitrogens. Same thing here. Remember we said glutamate's going to be our storage form. Glutamine has 2. Again, look at this. So we have 2 molecules of ammonia. We can't get rid of it immediately. What do? Well, we want it out of the circulation. So we're going to transfer 1 to glutamate. We could transfer 2, and then we get glutamine. So those nitrogens are up on glutamine, but we've effectively gotten rid of 2 molecules of ammonia. So it's not in circulation. But again, you're eventually going to want to get it down to alpha ketoglutarate, get it through the Krebs cycle. So you may end up making ammonia. Ideally, you'll be able to get that to the liver, form urea, and eliminate it through the urine. Okay, same thing here. Glutamate will store it. If you have to store the ammonia, you can get it to glutamate, or get rid of it to alpha ketoglutarate, you have to make ammonia again. So these are all a common theme here, and I'll show you the clinical repercussions here in a second. So again, ammonia is neurotoxic. So you have to convert it from the peripheral tissues to the non-toxic form. This is why we use glutamine and alanine to do that, to get it back to the liver. So these are the 3 processes we talked about. You get it from alanine, glutamate, glutamine, get it to the liver so you can make urea, so you can get rid of it. Now, like I said earlier, if you have some sort of liver problem, you may be sending too much urea out in circulation. You may not be able to store it as well until you can get it to the kidney. So we look here. So look at the nitrogens here. This is not charged. So it's not going to affect the acid-base balance as well. So again, you make this in the liver and you get it to the kidneys. Unfortunately, this is one that I don't know if I really remembered this whole thing. I kind of just remembered the diseases and what went with it. Because like there were a few things. Hold on, I'll show you in a second. And this is just, I thought the first aid picture looked a little bit better than this, but maybe just remember the enzymes that correlate with which of the diseases. So these correlate with the different steps, but if you want to draw that out, that'd probably be good. Yeah, so these are different problems that are going to basically cause hyperammonemia, right? These problems cause a buildup, so you're not going to be able to get through the cycle, and you're going to have too much urea formation or ammonia formation. Sorry. So, and this is kind of what I remembered actually. So, like most of them are very similar. I believe CPS1 deficiency is the most common, so type 1. And also OTC, hyperammonemia type 2, is also ornithine transcarbamylase deficiency. So you get increased ortic aciduria. So like if I was looking at this and I was like, what stands out? This would be something that stands out, right? The ortic aciduria, that will differentiate it from the rest because everything else is pretty similar. Also, arginemia does not cause hyperammonemia. It's the only one that doesn't. So if you don't want to draw out that pathway, maybe just knowing these enzymatic deficiencies, you may be good to go. Okay, this is very important. This came up on the test as well. How do you treat this? Well, you can give medication to kind of mop up the extra ammonia, right? So you can give phenylbutyrate. I remember somewhere him talking about phenylacetate too. So you can use these interchangeably because it's actually converted. So that's a medication. You can also get benzoic acid. These will work to mop up all that ammonia and get it out of your system. So this is basically the problem. Neurotoxic, the ammonia, and so you get cerebral edema. This is swelling. Look, you could barely see this fissure in the middle. So there's a lot of swelling. You can't appreciate the gyri. I'll get into that later. But basically, this is a lot of swelling going on. CT scan, obviously the bone's lit up, right? So cerebral edema is the problem. But what is causing that? Well, this here. Remember, we said glutamate will hold 1 nitrogen. Glutamine will hold 2 nitrogens. So if you have a bunch of ammonia running around in circulation, you're going to have to store it until you can get it out of the brain, right? Or get it back into the liver. So these glutamine levels are going to be really high. So you're going to get this osmotic imbalance, right? With all these particulates around, you're going to drag water with it. So the water is going to cause this osmotic imbalance, and you're going to get cerebral edema, right? So that's basically what you want to get out of that. So let's talk about these. Like I was mentioning before, figure out for these amino acid problems, what stands out about it. So between this and the other amino acid problems. So phenylalanine hydroxylase is PKU deficiency, classically known as type R. The classic PKU is PKU1. So you're going to get this mousy odor. What does mousy odor smell like? I really don't know, but it's mousy, whatever that means. I think it's like foul smelling or something. I don't know. You could look it up, but I've never smelled anything mousy, so I don't know. Right. So this is the problem here, but they'll probably tell you this, this mousy odor. Now, PKU is one of the things that they test for right after birth, and that is because if you take the phenylalanine out of their diet as early as possible, they don't have long-term effects. So if it's not picked up until later, they may have neurological problems. But the idea is if you could take this out of there, if you can identify it and make sure they don't ingest a lot of phenylalanine, then hopefully they can grow properly. So this is a good example of that. This little girl had it restricted in her diet early, so she didn't have some of the neurologic problems that that form if you don't catch it early. So the main thing is avoidance. Seropterin, it was also one, I don't know if that's really high yield, but it's a synthetic form of tetrahydrobiopterin, which is BH4. So you can use that as well. Maybe just make note of that. So importantly, increased phenylalanine is a teratogen, right? So if a mom has, or if a pregnant woman has PKU and she is not monitoring her phenylalanine levels, this can be transferred to the fetus and it can have teratogenic properties, okay? So it's important that the mom be aware that she needs to keep the phenylalanine to a low level, which she should be anyway, because if she has the disease, but even if she's a carrier, it would be important as well. Okay, so alkaptonuria, so what stands out about this? The urine turns dark on standing, okay? It's like the oxidation reduction reaction. So you wanna know the enzyme here, homogenistic acid oxidase. This will cause alkaptonuria. I don't know if this is one of the things I would bother drawing out. I would just know this is caused by this reaction right here. This is the enzyme. So what stands out about it? Again, darkening of the urine on standing. You can also get discoloration of the cartilage, severe arthritis. Look at the joints here. That's not good. Okay, and again, you get excess homogenistic acid because you don't have the enzyme. Tyrosinemia is not, I think on standing is because I think it's oxidized on standing. I'm not 100% sure. Yeah, oxidation. Yeah, I believe that's what happens. So it turns dark. So tyrosinemia is super high yield, but you should know this. Again, what stands out about this, apparently smells are like this theme here. So it has a cabbage-like smell. I don't even know what that smells like, but it smells like cabbage. It doesn't sound good, but okay. It can lead to liver and kidney damage. Another one, this one probably smells a little bit better. Maple syrup, urine disease. So you have this branch chain alpha keto acid dehydrogenase. So if this sounds familiar, this was that third enzyme we talked about previously that uses the TLC for Nancy, those 5 coenzymes, thymine, TL, lipoic acid, CoA, FAD, and NAD. So this is that third enzyme we talked about. So for this, you can end up getting, so make sure you know the enzyme. B1 is one of the 5 cofactors you need. You can end up getting ketosis too, but I imagine they would tell you some sort of sweet smelling the urine. Okay, they can get neurological signs. Remember, I did mention that ketosis is a problem, and again, sweet smelling maple syrup urine. Wish they named everything that easily. Okay, so treatment. Basically what I wanted to point out with this, these were the 3 enzymes we talked about. Pyruvate dehydrogenase, PDH, alpha ketoglutarate dehydrogenase, and now this branch chain alpha keto acid. These are the 3 that use the 5 coenzyme, okay? So if you're lacking in any of these, that can cause a problem. Make sure you know that. Okay, so for, what are we talking about? Oh, this is the other one. So methylmalonyl CoA mutase, it uses B12, right? So one of the 2 places in the body that uses B12 as a cofactor. But this is that, when you talk about breaking down valine, isoleucine, and methionine, or these odd chain fatty acids, right? So this is the process. So this, the 3 carbon, or 3 carbon groups, yes. Propionyl CoA. Remember, any sort of carboxylase is going to use biotin. And then this is the step that's a problem. If you have B12 deficiency, it's also problematic here. So this is how these kids present. A lot of neurologic manifestations, encephalopathy. Again, this wasn't, I don't remember this being tested, but I just want to put all the clinical stuff in year 2. Okay, so homocystinuria, that's right, sorry, the thing's covering it. So cystathione beta synthase is the problem here. So you get buildup of homocysteine, which is toxic in the body. So this will cause homocystinuria. This does cause cartilage problems. So if you see this presentation, looks a lot like Marfan's to me, and so do the symptoms because it's a cartilage problem, right? Dislocation of the lens, subluxation, similar to Marfan's. Skeletal abnormalities, look at the long limbs and stuff. So premature arterial disease. So you want to know this. Sometimes you can give PLP or vitamin B6 for treatment there. So this is a good one to know the symptoms for. All right, so I did mention this during the break. I found Dr. Bandelow's lectures a little all over the place. It was kind of difficult to pick up on, so I was kind of worried about his stuff going into the exam, but I was pleasantly surprised that a lot of his questions are straightforward. So that being said, some stuff I left out on purpose just because I wanted to focus on the stuff that he tested. Okay, so the enteric nervous system, basically it runs independentally of the ANS, right? So you have, they work synergistically, but they are independent of each other. So you have myenteric plexus or Auerbach's plexus, and you have the submucosal plexus. I remember myenteric, it sounds like muscle, my is like muscle, and it's in between the 2 layers of the muscle in the GI tract, the longitudinal and the transverse layer. So we have sympathetics, obviously that's our rest and digest, and then sympathetics, which is more running from a bear, so it's inhibitory. So myenteric is going to be worried primarily with motility. Submucosal plexus or Meyer's plexus is worried about secretion. So it's easy to remember, M goes with M, S goes with S, right? Secretion versus motility. All right, Lindsey mentioned this toxic megacolon. This is kind of the image they would give you right here. So this looks like a barium swallow. Or maybe retrograde. Yeah, it looks retrograde here. So you can see this aganglionic segment, failure of neural crest cell migration, right? And that's the problem here. So at this point, you don't have peristalsis here, and you can see that it kind of just stops right there. So without peristalsis, you really can't push anything down. So this is that portion that is the megacolon. Okay, so when we break down the different areas, we have neurocrine peptides, which are neural origin, endocrine, which will be sent through the bloodstream, and paracrine, which is cell to cell. I wanted to put this in for completeness sake. If you have a general understanding of these, particularly this slide, you should be okay for the question you can ask. Unfortunately, it does get a little confusing when you talk about increasing and all the increasing and decreasing, whether acid feeds back or feeds forward and all that stuff. But try to have a general understanding of what's going on and the cells that are produced, because those are the questions he asked. CC came up a bunch of times, maybe 3 or 4 questions. So know everything that goes along with that. Gastrin's going to be involved with the parietal cells, going to be involved with acid secretion. We'll talk about somatostatin a little bit. Secretin is kind of opposite of gastrin, where gastrin is going to produce acid, secretin is going to produce bicarb. You would expect acid to be produced in the stomach. You would expect bicarb to be produced in the duodenum. So when that acid chyme or that bolus goes through and it's very acidic, it gets into the duodenum, there's a lot of bicarb to kind of neutralize that charge. Let's see, not as important, but this was in first aid as well. So we'll talk about motilin with the migrating motor complexes in a second. Yeah, and we'll talk about these. Yeah, and this is some other stuff. Lindsey mentioned intrinsic factor. Remember, you need that. Intrinsic factor will bind B12 in the stomach and it will transfer it to the terminal ileum where B12 is absorbed. The question they like to ask is if you have a terminal ileal resection, you cut it out, you're gonna have, you can't absorb B12, right? And you can't absorb fat soluble vitamins either. So that's a good clinical correlation there. If you have some sort of autoimmune problem, we actually just covered this in immuno, and you have this parietal cell destruction, you also will have some sort of B12 deficiency. If y'all remember, B12 and folate will cause that megaloblastic anemia. So you can differentiate B12 from folate deficiency because B12 will cause neurologic problems. So if they ask you something in regards to this and there's some sort of neurologic problems, a lot of times that has to do with B12. But that may be a little bit much for now, but just in case you wanted to know. All right, I like this picture from first aid. Kind of tells you where the different cells are, whether we're up in the fundus or this region here, or down by the antrum here, where the parietal cells are. So the food comes in, you get that acid, and then, like I said, the eye cells over here in the duodenum will secrete CCK. The secretin, the bicarb, will be secreted here. And remember, when you looked at histologically in those deep crypts, the pepsinogen is going to be down here. The chief cells will be down here. That will secrete pepsinogen. Okay, so I'm not going to read all these slides to you, but these kind of go just a little bit more elaboration from what from the first aid slides I just had. So gastrin is going to be coming from the parietal cells. It's going to be involved with acid secretion. The acid will actually also feed back to inhibit extra acid. It's just a negative feedback mechanism if there's too much acid. Yeah. CCK, super important. So you want to know everything it does. It's going to contract the gallbladder and also relax the sphincter of Oddi. It'll help with pancreatic secretion of bicarb, stuff like that. It'll also inhibit gastric emptying along with somatostatin. So it allows more time for digestion, kind of like the food like soak in the stomach juices and stuff, right? You don't want it to transfer too fast. Secretin, I mentioned, it's opposite of gastrin, right? So it's going to be involved with bicarb secretion from the duodenum, or it's going to signal the secretin's going to signal the pancreas to release it. Don't get that confused. So secretin from the S cells will go to the pancreas and signal the bicarb to secrete. To the pancreas to secrete bicarb. GIP and GLP, you should know that, they both enhance insulin secretion. So even though it's a glucagon-like peptide, that's kind of seems backwards, but just know that the Gs will help with insulin secretion. And then there's paracrine. Remember, those are going to be cell to cell. So these are going to be very, very restricted primarily to the stomach. So again, somatostatin is going to slow gastric emptying, and it kind of just allows the food to kind of just soak there for a little bit and it to break down. Histamine as well. So histamine, this is the same histamine that you have an allergic reaction with. Just depending on where in the body it is, it has different receptors. So I believe these are the H2 receptors that are in in the stomach. So those will help with gastric acid secretion. Okay, so you can actually take histamine inhibitors or antihistamines, where some antihistamines you would take for allergies, some antihistamines you could actually take for gastric acid secretion. Same histamine, just a different receptor. Okay, so the neural ones, the neural peptides, you have VIP, you should know that. It's similar to secretin, so it'll help to limit the acidification. Gastrin releasing peptide as well. This is neurocrine because it comes from the vagus nerve. Even that cephalic phase where you're thinking about food, right? You do get a release of from the vagus nerve of modulators of digestion, right? And then the encalphins, I don't know if that's super important, but again, maybe just knowing that they're related to the neurocrine system. I remember there actually being a question on the test. So like G cells make gastrin and this CCKB receptor. I didn't think it was important, but it ended up showing up. Whereas histamine is the H2 receptors. That's where you could take antihistamines for like reflux. And vagus obviously will be the muscarinic receptor, right? M3 specifically. So acetylcholine. So make sure you know these and what they correlate to. Now, when we talk about leptin versus ghrelin, remember leptin is going to be released from your adipocytes, from your fat cells, to tell you to stop eating, right? Staying satiety, right? Where ghrelin, I think of like grr, like the stomach growling, right? So it's like a hunger signal, right? So grr, you're hungry, right? So they're opposite of each other. But also know that insulin, which makes sense because you're in a well-fed state, and GLP will also inhibit appetite. So good to know those. Now, when we talk about this, we're looking at here, this is a pressure wave, okay? So what you can see here is this correlates to right here. So you tell the patient to swallow at this point. So you get a sharp pressure right here. And then as the bolus moves down, you get a lax in pressure, right? So an increased pressure. And what that's going to do is help to move the bolus all the way down the esophagus. That's the idea. And you can see right at the end, which is different, right? This relaxation in pressure, right? So this spike in pressure will help to move the bolus all the way down. But once you get to the lower esophageal sphincter, you want to have a relaxation in pressure. So the sphincter will relax and allow the food to enter the stomach. So you may get a question on manometry. Just understand it's pressure, right? You want the bolus to move unidirectionally. You don't want it coming up, right? So you kind of want to have contraction to like behind the food, right? To kind of move it down. That's the whole idea here. Okay. So, right, and then there's different processes, right? So you have this segmental contraction. So that's just going to help to like mix and churn the food. It helps with digestion, right? So you kind of just it's not moving down the digestive tract. Peristalsis is going to be the process of moving down. So these work together to kind of like make the food bolus and kind of keep it consistent, but also the peristalsis will help them move it down. So keep these separate in your head. Okay, and then understand that tonic contractions are going to be like sphincters, right? You want those to be tonically contracted. You want them to only relax when something has to go through. Phasic contraction, those are going to be like peristalsis, right? It goes through phase, it goes through waves to go all the way through the GI tract. So it helps to make this unidirectional flow of the food. So GI motility, acetylcholine, again, parasympathetic, right? So you're going to get these action potentials. We'll talk about the slow wave stuff in a second. But just, I think I wanted to point this out. Yeah, these are just the opposite. Where parasympathetic or acetylcholine will help to increase the action potentials, VIP and nitric oxide will decrease them. So this is just one of those things that stands out. Now, the migrating motor complex is going to be the peristalsis or the movement of the gut when you're in a fasting state, okay? So it helps to move things. So a little bit of food that's here, or you don't want bacteria to be static in there. So it's gonna be this migrating motor complex will help to just move, but just make sure you understand that this is not during the fed state. Okay, this is just when you're fasting, but it wants to move. You don't want stasis of anything in the gut. Okay, and then what you want to know is motilin is going to be the hormone that helps with this migrating motor complex. So again, this is in between the feeding states. You're in the fasting state, and it just kind of helps move whatever is in there, just move it around and stuff. All right, so these slow waves, it's like this weird thing. It's like kind of hard to grasp, but first off, you need to know that these interstitial cells of Cahill, I don't know if I'm pronouncing that right, but so these are going to help with these slow waves. So basically, if you think of like an action potential here, right, and these slow waves are going to just be going right underneath it, right? So they're just waiting for a little up in the action potential or a little bump up through threshold to kind of make the wave. So they're kind of just these, like they say, they're slow waves. I think I have a better picture, yeah, right here. So look, so these are the slow waves. They're just kind of going, minding their own business until the action potential hits a threshold. So they're just ready. And so it just takes a little bit of an action potential. So it's just this light firing that brings a slow wave above threshold, okay? So that's the idea when you're moving food, when you're going through peristalsis, even segmentation. These slow waves just need to be bumped up a little bit, and then you get this muscle contraction. So it kind of keeps them prepped, like right on the verge of firing until you get action potential. And depending on, yeah, I do believe it. Yeah, it would be summation, and you can see the summation here, right? So you see this small firing of the action potential here, and then with the summation here, you can see that here for greater. So it kind of makes this nice, elegant. It's not too much. It's kind of just like a slow peristalsis that goes through. So these slow waves are always ready to go. That's how I understand it. Now let's look at the difference here. So remember, if you're in the interdigestive or the fasting state, these migrating motor complexes are going to be going just to keep your gut clean, keep things moving a little bit. And then in the fed state, you're going to go through the peristalsis and the segmentation. So you can see the difference here, what's going on. This is more of a controlled process where the migrating motor complex under the control of motilin is kind of going. Okay, and so primarily the fundus, we talk about this reservoir function. It can kind of hold food up there, all the while the food that's down in the antrum area via this peristaltic wave, you could start getting some of the acid and the chyme or chyme, however you pronounce it, in here, and you could start digesting it and then move it into the duodenum. So know that the fundus is the region that's going to be more of the reservoir area. Now, when we get to the large intestine, you're basically worried about these mass movements. By this point, the food has been segmented. It's kind of contracted, getting all of the nutrition out of it as you can. The only thing you're really worried about in the large intestine is pulling back water, maybe the potassium balance. So you're going to, as you move it across, you're going to move this mass movement here, and you see that here. So these large segments of food is going to be moved, but they call it this mass movement effect, right? And haustral shuttling. So the haustra are these different segments you can see here. Make sure you can identify those on a barium study as well. So you get this mass movement across. And what you can see is as you move down the GI tract, the transit time correlates with the slow wave time. So that you do not have slow waves in the esophagus, you do not have slow waves in the rectum. But as you move through, you can see that the transit time and the slow waves correlate, right? So whereas in the distal stomach, transit time's 1 to 3 hours, slow waves are pretty frequent, right? You move to the small intestine, 7 to 9 hours, slow waves are a little less frequent. Large intestine, again, same principle here, right? And depending on, yeah, so you could just see that here. And again, once you get to the rectum, you don't have slow waves. All right, I wanted to point this out. These are some of those reflexes. So you can think of it like this. If you bring food into the stomach, these are a way of the stomach telling the lower aspect of the GI tract, like food's coming in, so food's gonna soon have to come out, right? So that's the whole idea. What they talked about on our test was the gastrocolon reflex. So basically, when you bring food into the stomach has a reflex to tell the colon that we're bringing more food in. So very soon, more food's gonna have to come out, right? So this is just a reflex so you don't get constipation. These are just the sphincters. If you want to take note of these are, I mean, you should know these by now. but these are where they're located. Remember, they're tonically contracted versus those phasic contractions of peristalsis. All right, so this is the salivary secretion. So it starts out isotonic, and as you move down to where the salivation exits in the mouth, you're going to start bringing in sodium and chloride. So sodium chloride is being reabsorbed, so it's not going to be out in saliva. And the hypotonic, you're going to be getting rid of potassium and bicarb. So think about it like this. If this flow, for whatever reason, is fast, then you're not going to have time to properly pull the sodium chloride back, okay? So you're going to have an isotonic solution. If this process is slower, you're going to have time to bring the sodium chloride back. You're going to have time to put the bicarb and the potassium in the fluid, and all the while making it hypotonic, right? More watery, right? So that's the idea here. And this is more confusing than it needs to be. If these are flipped, if it's isotonic, that means the flow rate's fast, right? So if it's fast over here, you're getting rid of a lot of stuff. If it's slow over here, the flow rate, you tend to be in more of an isotonic state, right? So that's just the idea here. But if they give you a question and they talk about this isotonic or this speed at which the saliva is being secreted, you can determine about this. Ideally, you want it to take a little while so you can get hypotonic, so you can pull back the sodium chloride. That's all that's about. Okay, this is just the process in the stomach, or with the gastric acid formation. So it's worth a read. The idea is you're going to secrete, as you would expect, acid, and then you get bicarb secretion. This alkaline tie is related to the bicarb that's secreted. This increased bicarb you can actually measure it in venous blood, right? And that's just because of the increased bicarb. So just the process here that goes with the gastric acid secretion. Just wanted to point out, so the cephalic phase, like I mentioned earlier, vagus nerve, acetylcholine, that's when you're thinking about food, you start salivating, stuff like that. Gastric is primarily going to be modulated by the enteric nervous system. So again, you still have acetylcholine, but also gastrin releasing that acid. And then intestinal, you start having amino acids as well. So these are kind of opposite factors, right? So the hydrogen is going to be counteracted by the bicarb and the mucus. So you want to have a good balance here so you don't get any sort of ulceration in the stomach or reflux or GERD. So some of the medications you can use, these are omeprazole is Prilosec. So you can get that. That's a proton pump inhibitor. So this hydrogen potassium ADPase. Histamine blockers or antihistamine, cimetidine, ranitidine. These are H2. Remember, H2 is in the stomach. H1 is what you're going to think about with anaphylaxis. And then you also have muscarinic antagonists. Ideally, you wouldn't take atropine for something like this, but these two are probably the ones you want to keep in mind. And then the pancreas, right? So remember, these cells are going to be coming from both secretin and CCK will be coming from the duodenum, and they're going to signal the pancreas to secrete bicarb. Along with these enzymes, remember there's zymogens, they're pre-proteins, right? They need to be cleaved to be activated. And I'll get into that in a second. But these are cells from the duodenum are going to signal to the pancreas to start secreting bicarb to counteract all that acid that's forming. Let's see. I don't know why I put this in here. I guess just to solidify the idea that this bicarb is going to be coming from the pancreas, right? So this bicarb chloride, do they talk about? Oh, they do. Okay, yeah. So CFTR, this is the problem here. Remember, with cystic fibrosis, the chloride, the chloride ions can't go across. The CFTR channel is messed up. If you can't get the chloride across and make this negative gradient, you're not going to pull sodium across to counteract it. Okay, so you have this negative charge. Sodium's going to follow to counteract it. And as we know, sodium goes with water. Water goes with sodium. So they're going to be very dehydrated in the sense that this mucus is going to build up, okay? Because they don't have this water, because they can't bring chloride, so they can't bring sodium, so they can't bring water. They get mucus buildup, they get ductal blockage there, so they don't get this bicarb out. They have problems absorbing some of the fat-soluble vitamins, amongst other things. Okay, now, this is similar to the salivary secretion. So at low flow rates, you're not going to be able to modulate it very well. But at high flow rates, ideally, you're going to get rid of a lot more bicarb, and you're going to bring back the potassium and the chloride. Okay, so ideal situation if you have, wait. Okay, so I said that backwards. At low flow rates, you're going to have primarily sodium chloride, okay, up here. At high flow rates, like after ingestion of food, and you get the signaling from secretin, you're going to get a lot more bicarb, okay? So you can see that here. Just make sure you have your axes and don't get it confused like I just did, right? So high flow versus low flow. Okay, and then again, we kind of talked about this. So that CFTR, this chloride that goes across, remember that the sodium is going to go with it, so water is going to go with it. So if this is messed up, they get a lot of blockage. So again, this is a clinical correlation that can correlate, that talks about this. So make sure you understand the idea of that. Okay, so again, this just reiterates the I cells, the S cells, they're going to feed into the CCK secretin, and you're going to bring in some enzymes, increase enzyme secretion. And the S cells, the secretin is going to be primarily involved with bicarb. And this vagovagal reflex, which is cephalic, is going to help with acetylcholine, and it'll help with the secretion as well. So in the intestine, remember small intestine, you're primarily going to be secreting bicarb, and large intestine, more importantly, we're going to be dealing with bicarb and potassium. And when we look at some of the causes of diarrhea, this is going to be come into effect. We'll get to that in a second. Oh, right here. So osmotic diarrhea is going to be more watery, and then secretory is going to be mucus or even blood buildup. Can happen from sort of toxins, something like cholera can cause that. So you want to differentiate between these two. But one of the big problems with diarrhea is you're going to get rid of a lot of potassium. So you're going to end up with hypokalemia. You know, that has a lot of effects through the body, including the heart and whatnot, and action potentials. So potassium's the one thing, but also bicarb. So anything with diarrhea, you're worried about bicarb. So if you lose bicarb, you get metabolic acidosis. You lose potassium, you could end up with hypokalemia, leading to cardiac arrhythmias, right? That's pretty bad, pretty serious, but it could happen. And then obviously, if it's osmotic, you're losing a lot of water. You get hypotensive, circulatory collapse. All right, let's finish it up with digestion and absorption. A lot of this from Dr. Trotz is elaborating or kind of summing up the stuff from Dr. Bandelow. So you want to kind of know where in the body different things work. So carbohydrate digestion, you're going to be primarily involved with salivary alpha amylase. Okay, so these 1,4-glycosidic bonds, you're going to break down starch, basically. That's what you do in the liver. In the stomach, we talked about gastrin with parietal cells, and then chief cells are going to secrete pepsinogen, right? Now, so once we release secretin, again, like Dr. Bandelow was explaining, the secretin and CCK is going to help with contraction of the gallbladder, secretion of these pancreatic enzymes, this bicarb, and the enzymes as well. So this will bring this acidic pH back to neutral pH. This is really important. You're going to want to remember all the effects of CCK, right? The bile, the enteropeptidases in the duodenum, because these will help to cleave and activate those different enzymes, or those different proteins or enzymes, yeah. And then pancreatic enzymes, or the zymogens, are the pre-proteins that need to be cleaved to be activated. All right, so the enteropeptidase is going to control this activation step. So the idea is once you cleave trypsinogen to trypsin, you could kind of cleave everything else. So this is activation from trypsinogen to trypsin will further activate trypsin cleavage, and it will also facilitate the cleavage of the rest of these proteins as well, or these enzymes, excuse me. So that's the idea here. Once you get trypsin going, you can get everything else going. And then this is just a big picture slide. You could read this. It kind of says like where everything goes. Here we go. You can see trypsinogen, inactive zymogen. Enteropeptidase cleaves trypsinogen. Once trypsin is activated, everything goes, right? Everything gets cleaved. So this is the limiting step involved in trypsin activation, or the enzymatic activation. Again, this is for carbohydrate digestion. Remember, salivary alpha amylase. We're going to worry about starches. Stomach, you're not worried about carbohydrates in the stomach, but you could read through this. We talked about brush border cells with lactose. Remember, sometimes you get lactase deficiency with these. And then we get lipid digestion. When you think lipids and lipid digestion, you should think bile salts. Okay, those are going to help with the digestion. Now, excuse me, when we talk about bile, the transportation of free cholesterol, that's going to be the big thing. So any sort of increased cholesterol, you can end up with bile stones, but bile salts are going to help to transfer the cholesterol or solidify or solubilize it. You could also, it also is an emulsifier, so it helps with absorption and breakdown of lipids. I wanted to mention orlistat. It is used for weight loss. So basically you don't absorb the fats. You don't start breaking them down to triglycerides. So they just run through your GI tract and they end up in the feces. So orlistat is a drug that works with weight loss. Bile salts also make these mixed micelles. They kind of surround the lipids. So it helps with mucosal absorption. So this is the main functions of bile: transport, delivery, formation, and release. But you could read through this. Excuse me one second. All right. So, but yeah, these are important steps because you can remember if there's any sort of decrease in bile acids or increase in cholesterol, we can have problems with gallstones. Steatorrhea, remember the bile is absorbed. It is involved with lipid digestion. So if you don't have proper biles or you're not conjugating them as well, you can get fatty stools or foul-smelling stools. Cystic fibrosis is one of the clinical correlates that has to do with this as well. Obviously, cirrhosis will work in the same fashion. If the liver's not working, you can't properly make bile salts. We talked about this before. The gallstones, cholesterol forms, stones form in the gallbladder. They precipitate out. And there's a lot of problems that can be caused by this. Even trypsin can be prematurely activated if this is the case, because if the gallstones block that pancreatic duct, like you can see here, trypsin will be activated in the pancreas. So it'll actually eat up the pancreas. It's supposed to get through the pancreas to the duodenum before it's activated. But if it's activated prior to in the pancreas, you can get autodigestion of the pancreas. Now, there's a little chart coming up, primary versus secondary bile acids, but basically these bile salts are going to emulsify these dietary lipids, right? And then in the terminal ileum, these bile salts will be deconjugated by bacteria and back to taurine and glycine. Okay, so these bile acids are made into bile salts by adding the glycine and taurine to them. There's a little, like I said, there's a little chart coming up that'll help outline this. And these primary bile acids originally synthesized in the hepatocytes, like I said, glycine and taurines are added, then they form bile salts, and then they could work properly from that point. And then you could see that here. So cholic acid, add glycine to it, and you could see here, or taurines added to it. So glycocholic, right, with the gly on the cholic, glycocholic, right, taurine with the taurine. So these go from bile acids to the more active form bile salts. They go through the gut, and then the bacteria will convert them back into bile acids. Okay. Kind of talked about that. Okay, and then this is good. So even if, the whole bile process could be a couple lectures long. So if you just want to break it down like this and just know the nomenclature or the names here, what's primary bile salts, secondary, you could see this here. So that'll help you, because Dr. Trotz could simply ask a question, what is an example of a bile salt? Something like that. So make sure you know that. And we are done. We will post the recording later, and I'll add Lindsey's slides to mine, and we'll post that.
If you guys want, I wasn't able to fix the issue, but I can scroll through that digestive glands lecture with you guys and point out some things you need to look at, because that is a very high yield lecture. I was looking through the PDF, and there's a lot of stuff on there that they do want you to look at. So share screen. Here we go. So I'm just going to quickly scroll through here, explain a couple things that I've heard people get really confused on and things that you really do need to pay attention to. So this is from mine. I don't have access to yours, so just letting you guys know that. So these are my slides, so I don't know if they're different from yours. Okay, the first couple slides, this is really stuff that you have already talked about in all of this stuff, so I wouldn't spend too much time unless there's something on here you need to review. Structural organization, the biggest thing is the porta hepatis. They can ask multiple styles of questions, so you need to know what this is. So hepatic artery, portal vein, hepatic ducts. You need to know what that is. But all of these, this is in subsequent slides. Blood supply to the liver. You have the hepatic artery, you have the portal vein, and then you have the sinusoids that are the distributing throughout. Now the parenchyma. They are going to go through these. You have a structural lobule and you have a classic lobule. They're oriented differently. They have different functions. So make sure you understand this. I've heard a lot of people get really confused about this. So the classic lobule is centered around the central vein. So if you see the central vein, that is in the center of the classic lobule. And that says portal area contains the branches of the hepatic artery, portal vein, bile duct, and lymphatic vessel. So that's going to be at your corners, essentially. So it's centered around the central vein, and then you have these portal areas that are going to be outside. That defines the boundaries of the classic lobule. So again, this is it. And then you see the sinusoids that are going inward. So this is the central vein. You have the portal areas and the sinusoids going in. You do need to know the boundaries here. So you have the neighboring hepatocytes, and then you see where you have the basal surface is adjacent to the sinusoids. This space of Dis is important. So microvilli assisting transfer of materials to and from hepatocytes. So remember where this hepatocyte is. These pictures always kind of confuse me because I couldn't orient. It took me a while to orient myself. So make sure you spend a little time and know what all of these are. But Kupffer cell, that's a big one, lined by Kupffer cell. So phagocytic cells. Remember, a Kupffer cell is just the name of the macrophage in the liver. So macrophages have different names in different areas. So a Kupffer cell is just a macrophage in this area. That's all that is. Space of Dis. Again, please know this, know what it is. This Ido cell is going to be really high yield. So you have the sinusoid, this Ido cell, and then the hepatocyte right next to it. Now, functional lobule. So the classic lobule is really easy to see because you have the central vein in the middle, and then you have the portal areas on the other sides. But the functional is a triangle made by the central veins, and then you have portal area in the middle. This is going to define the zones. So we'll talk about that in a sec. The zones are really important with respect to transfer of material. So biliary tree and flow of bile. You need to understand the canaliculi and in its association with the flow of bile. So you have tight junctions between the adjacent hepatocytes containing short, blunt microvilli. And this receives the exocrine secretion of the bile. And they convey it, so it sends it to the canals of Herring. I don't think cholangiocytes was a huge thing they did with us, but the canaliculi was definitely something that they focused on in our exam. Come on. So when I said the areas, the functional zones, this is what I meant. So zone 1 is the first to be exposed to entering blood, and the first to show changes after bile stasis. And then zone 3 is last to be exposed. So you see the central lobule, you have these portal zones in the middle, and this functional area, this functional lobule that is formed by the edges of the triangle are the central lobule, central vein. This is where you see the zones. And so 1 is going to be closest to the portal area, and then it goes out when you get to the central vein. And so you need to know the first exposed, the last exposed, because it says right here, consider implications of toxemia or hypoxemia, or really anything associated with the distribution of nutrients through this area. And this is another clinical correlate that goes directly hand in hand with this. So you are going to see in zone 3, you have a lot of vacuoles. So you have extensive lipid accumulation here. So they can put this on your exam and you see this lipid accumulation here, and that can tell you about congestive heart failure. So this is just putting it all together in the same, into the same slide. But that was the biggest stuff. Fatty liver, this is just increase in the lipid droplets. I don't remember them being hitting that too hard. Liver cirrhosis is a big one. And then they get into gallbladder. I think they put this on our exam. I can't remember how they talked about it histologically though. Do you remember, Brady?
Didn't they say something about false lumens? Isn't that gallbladder?
Yes.
They form those false lumens. Yeah, I think that's what they talked about. Maybe on this slide. Yeah, those folds.
Yeah. I spent a lot of time trying to understand this, but they didn't hit it as hard as I thought they were going to hit it. Like this lecture is pretty high yield, but it's high yield in the sense that there are a lot of areas they can take like one question from, 1 or 2 questions, but they will pull from this. There was another big one that I wanted to focus on. Oh, here, yes. You need to be able to look at the pancreas and know if they point to a cell, know which one it is based off where it is. Because remember, alpha cells and the beta cells, they are going to be distributed differently. So one is going to be kind of in the center, and then one is going to be kind of dispersed at the periphery. So you need to know that. So alpha cells at the periphery, and then beta is going to be more in the middle. So that was something that you needed to know. But the rest of this on here, histologically, I don't think they hit very hard. But know the functional lobules, the transition zones, that was high yield. And then the islets, that was big. But histologically speaking, those were the biggest things in this lecture. Are there any questions? The difference between the large intestine and the small intestine. I got that confused too. It wasn't just Brady. The DLA with the glands is the salivary glands. The biggest thing to know is the difference between mixed and if it's, you know, mainly serous or mainly mucus, and then where those glands are. So I actually have it pulled up. No. Okay, I don't have it pulled up. Well, where it opens. So like one is like behind the second molar or something like that. But those, those are really the biggest things you need to know about the salivary glands. The difference between, I'll go back to that. Okay. So the difference between the large intestine and the small intestine. Come on. Stop it. Hold on, guys. There we go. Nope, that's not what. Oh, damn it, Lindsay. Okay. So it's really the appearance here. So if you look, I think it looks like a flower. That's what it looks like to me. It looks like a flower here. So this is the large intestine. So if you go back to the duodenum, these are more bigger groupings. So I kind of look at it, let me get so this looks like a bigger grouping to me right here with a bunch of individual ones in here. That's the best way that I can think of to remember it. And then if you go back to come on. Here, you see that more flowery appearance here. I like a lily in the large intestine. I like it. Drops in the duodenum. That's really good.
Thank you so much, Lindsay. Can you also differentiate by like the near the lumen, how they're different? Like in the small intestine, it was more like
yeah. So if you see this here, this is very close to the lumen here. That's what she was saying. But if you go back here, it's in an actually different place. So you see the submucosa here. You see these crypts of Langerhans, and you see the Brunner's glands down below in the submucosa. So it's actually a completely different place. So submucosal, Brunner's glands, if you come back here, it is much closer to the epithelium. That is a very good distinction.
Thank you so much.
But like in my head, when I saw it, I saw widespread. And I just, for some reason, because the plicae, of course, are really noticeable. And then the Peyer's patches are really noticeable. Be like, okay, that's a Peyer's patch. But these, I got it messed up in my head because it looks a little more dispersed.
So like differentiating the Brunner's glands from the large intestine, differentiating the rugae in the stomach from the villi in the small intestine, and differentiating the lymphatic nodules in the appendix from the Peyer's patches are really good examples of what you should know.
Thank you, guys.
And remember, they're going to use the slides that you have in your notes. They're not going to throw something weird at you, most likely, but you're most likely going to see what you have in your notes. And so make sure that you can pick out those distinguishing features and use that.
All right. Any other questions? If not, we'll go ahead and stop the recording. We'll see you guys next week and finish all the rest of the lectures before y'all's exam. Probably Saturday. We'll plan on that.
Yeah.
Later, guys.